Alright, here we go, you guys. I'm going to go through this lab on meiosis and hopefully give you guys a good idea of what you guys can do with this. So we've talked about mitosis, which we know happens in our body cells, and basically the outcome of mitosis is one cell going through cell division to make two cells that are identical. With meiosis, we know this is a little different. Okay, meiosis will produce ultimately 4 cells, and they are all genetically different. And this comes to our question. Our driving question for this lab is, how does the process of meiosis reduce the number of chromosomes in reproductive cells? And why is it important that it does reduce the number of chromosomes in there? Okay, and so to get a quick introduction, I'm not going to read through this. I will expect you guys to. But know that with sexual reproduction, two things basically happen. First, we're going to have cells that are being produced there. And second, fertilization will happen. And so it's really important to see these two steps in a model so that we can see and answer this question of why is it important that meiosis reduces those chromosomes in those reproductive cells. And so to get started, I love how the lab kind of shows this depiction of this karyotype, human karyotype. We know that humans have 23 pairs of chromosomes, okay? And how they kind of set up these karyotypes is they actually put the bigger ones, that'd be number 1, and as they go down, the smaller they get, the shorter they get, okay? And we also know that in each of our diploid cells, our cells that are diploid, meaning double the DNA, they actually have one. When we're looking at these chromosomes, one comes from our mother and the other one comes from our father. So one's maternal and the other one's paternal. The same for this one, maternal, paternal. And it could be vice versa, we don't know, right? And there's no way to really truly know which one came from which. You'd have to do some serious DNA testing to determine that. But anyways, every single one of our chromosomes, we got 1/2 of that chromosome pair from that homologous chromosome pair from our mother, and the other one from our father. Okay, and so we're going to model this today. So right here it tells us your task. And it says, meiosis is the process in which chromosomes are duplicated and then separated into 4 reproductive cells that have exactly half the number of chromosomes of the original organism. So, haploid. In addition, the process ensures that there are no pairs of chromosomes found in the reproductive cells. In other words, there is only 1 copy of each chromosome in reproductive cells instead of 2. Then during fertilization, a reproductive cell from a male sperm and a reproductive cell from a female, the egg, will fuse to form the embryo or zygote that has the same number of chromosomes as the original organism. Okay? This process happens in all animals, plants, and fungi that reproduce sexually. Your goal is to develop a model that explains what happens to the chromosomes. So this is your goal. Your goal is to develop a model that explains what happens to the chromosomes within a cell during each stage of meiosis. Again, there's our driving question. There's this part right here where it says getting started. Okay, and this will kind of dive us into what you guys will be expected to do. It says to answer the guiding question, you will need to develop a model that explains the process of meiosis. Okay, so we're going to actually use these beads, okay, to model meiosis, and you guys will need to do this and in different ways. Okay, so there's not just one way that you will do this. I'm going to show you one thing that you guys can do. Again, it does not need to be this way, and I'll show you some different examples, or give you some different examples of what you'll need to do. Okay. Anyways, the first thing that you're going to do is look at these. I have several pictures that I will have that you will need to look at. Okay, and each one's going to be pointing at a specific cell, and using this I want you guys to put them in order of going through meiosis, of how you would expect them to go. Okay, and it's not in order right now, so you'll have to go through those, and I want you to put them in order from the start of meiosis going through meiosis 1 and then going through meiosis 2 of what you would expect to see there. Okay, so this is the first part, is using those slides to make it kind of a model that way. And what you could do is you could actually even cut these out and place them in order so that you can use that. Okay, maybe number the back so I know what you're numbering, the sequence that you had them in. You could also just lay it out and show meiosis that way, because then what you're going to do is use these beads to model what is happening in these pictures. Okay, now with this model, your model, once complete, should be able to explain three things. First one, what happens to the chromosomes inside a cell as it goes through meiosis. 2, why reproductive cells have half the number of chromosomes of the individuals who produce them. And C, why there are no pairs of chromosomes in reproductive cells. Okay, so to make this valid, your model must be able to explain this. Okay, so it needs to be able to explain this, and I'm going to show you how I expect this to be done. So what I'm going to do is show you one model here, and how I'm going to do that is I'm going to get all crazy and wait till you guys see this. And I want to go back and see this so I can see what I'm doing. Okay. Alright, so I've got here a whiteboard. Okay. And you guys are more than welcome to use this type of thing if you'd like as well. And I will have some little laminate sheets out that you could model this, and you can use the markers that are there with it to use. Okay, so the first thing, I'm making this very simple. I'm only using one set of chromosomes. Okay, and we know that at the start we have our chromosomes like this, and like I said, one half of that homologous chromosome would come from our mother, and the other one would come from our father right here. Okay, so here I've got the cell drawn out, and I've got the nuclear membrane that's encompassing these chromosomes. Alright, so let me kind of model this. And I'm not going to go through all of meiosis, but I am going to kind of step through this, and you guys can see, maybe get some ideas, and hopefully also clarify the steps of meiosis as well. Okay, so first of all, we know that the first step of meiosis is interphase. An interphase can be broken into G1, S, and G2 phase, and it's during those phases that first the cell's going to enlarge to get big enough to support two cells. During the S phase, we're actually going to see these chromosomes duplicate and replicate themselves. Okay, and they are exact replicas. So we have this one as an exact replica of the paternal DNA, and here's the maternal DNA. We now have two exact replicas. Okay, and at this point, we're going to now move into prophase. Prophase 1. Now in meiosis 1, prophase 1 is very interesting. It's very different, and it's not the same as mitosis. So it's during prophase 1 that actually these homologous chromosomes will come together and form a tetrad, kind of like this. Okay, so they'll actually pair up, and if I had more chromosomes here, you would see more of these being formed. And so what we see happen here is during prophase, these line up, the nuclear membrane is gone at this point, or dissolved. Okay, and then we go to metaphase 1, and during metaphase 1, these guys are going to line up at the metaphase plate. Oh, I forgot one part. During prophase 1 is actually where a process called crossing over will occur. And what we see happen is these chromosomes, these homologous chromosomes will actually cross something like this, and they can actually cross up to, I've seen some literature say that it'll do 20 crosses. Okay, so it could zigzag back and forth through that. This is also a 3D model, and so these ones could be crossing over on this side too. If we had like 3D, right, these guys could be crossing over up here too. Okay, but so we don't confuse things, we're just going to show a crossing over event like this. Okay, during this time also we have these structures down here on each poles of the cell that will start to make these what these microtubules, okay? And they'll start to grow these out, and if we had more chromosomes, such as right here, let's say we had some chromosomes there, they would pair up right there, and let's say we have some more chromosomes there, so this one would pair up there, and right there. We would see the same thing on this side. These microtubules would form out from the centromeres, from this, the centrioles, I think is what they're called. I don't. Anyways, and would start to form on these centromeres. So these are centromeres. I used the wrong word, and I think these are centrioles that are in the two poles of the cell. Okay, and so these microtubules are going to attach. So that's mitosis 1. Metaphase 1, sorry. Mitosis 1. Metaphase 1. During anaphase 1, we'll see these start to split. Okay? And these microtubules are going to start to break and pull and tug and pull these cells apart. At this same point, these crosses are going to switch. Okay? So let's say, for example, that I have it switching right there. Let me hurry and do some switches here. Okay. Okay, so we have some crossing over that happened on both sides. These centromeres are going to split apart and pull these chromosomes to each end of the cell. Same thing here. We pull those to the end of the cell and pull these to the end of the cell. Okay? So now we have this. Anaphase 1 is now kind of concluded, and we're going to see telophase 1 start to occur, where we're going to see this cleavage event occur in animal cells and start to split apart. Okay? And so then that is the end of meiosis 1. Now we end up with two cells. Are they genetically identical? They are not. Okay? They are definitely different. They have unique DNA because of the crossing over that occurred. And then what's going to happen during meiosis 2 is each one of these cells would then split and go through meiosis cell division one more time. And the unique thing is in meiosis 2, crossing over would not occur. It's only in meiosis 1 during prophase 1. After that, this would not happen. And you would then split again, making two more cells in the four cells total in the process, all genetically different. Okay, so that kind of models that as well. Okay, so this is one way I just modeled meiosis. Oops, baby, wait. So I just modeled meiosis 1 and 2 using these beads. The thing is, I'm missing this really important part, because I explained it verbally, and now it worked. So what you guys want to do here is going back to our thing. Let me go back to this. Remember, there's three things that this needs to do. It should be able to explain what happens to the chromosomes inside of a cell as it goes through meiosis. B, why reproductive cells have half the number of chromosomes of the original, okay, and why there are no pairs of chromosomes in the reproductive cells. So this showed how these reproductive cells are formed. Again, showing you again. So these are each reproductive cells, whether it be sperm or egg that was produced. And they're all genetically different, and I kind of modeled some of this. Because I recorded this, this is a great model because I was able to explain what's happening here. I was able to show that each cell does not end up with, it only has one half of that, it's got one, it does not have pairs of chromosomes, it's only got one chromatid, okay? So I was able to show some of this verbally, and you guys are more than welcome to do that too if you want to record this and do that, okay? The thing is that whatever you turn in needs to be individual. I do not want a group of 4 people that turn in the same thing like we did with mitosis. So this is individual that you guys need to turn this in. Okay? And so you would want to do this individually, record it, and if you want to, you could explain. That's one way that you guys could do this. The second way that you could do this is, as I was going through this, take some pictures, okay? And then you can upload it and use those pictures in a PowerPoint slide, okay? And then use those PowerPoint slides to explain. So you could go to your OneDrive. Okay, so if I was like, for example, go to our PowerPoint here, and let's say that I wanted to use one of these pictures that I just took, not off the internet, because that's cheating. But let's say that I, hypothetically, this was a picture I took. I could then take this picture, make a new slide, maybe such as this, okay, add the picture, title what it is, Prophase 1, and explain what's happening there, okay? So write in words what's happening during Prophase 1, that during Prophase 1 crossing over happens, which is going to hit specifically the portion of how they are genetically different from each other, okay? And so anyways, that's another way that you could do that. I know some of you like to do those little still video things where you take pictures and then you stick them all together. You're more than welcome to do that too. And then, but if you do that, some way you need to give me an explanation, either paper or verbally, for that model. Okay, so the ultimate goal here is you're going to make a model, and then you're going to use that model to explain this. Okay, and so you need to somehow be able to explain that, either that be verbally that I can watch or listen to, or it could be written. Okay. Another option that you guys have is you could draw this out. So as you're modeling this, if you do not want to take pictures and do this electronically and you're more like the artsy type of person, you could get a piece of paper, and I've got paper up in those cupboards over there by the along the wall to the hallway. Anyways, and you could use one of those papers and actually draw what your model showed with the beads, every step, and then kind of explain what's going on there. Okay? So I'm going to leave that up to you guys, and we will work on this more when I come back. So it does not need to get done today, but please get a start on it. Don't waste time, and at least get go through this and model meiosis out, getting started on this process of making this model to answer our driving question. Okay? Anyways, I think that is good enough, and I'll let you guys get going there. And if you have any questions, please email me. My email is up on the board, as you guys know. Alright, if I can get this thing to shut down, we will talk to you on Friday.